Okay, my name is Refilwe. I also go by Tana, as Casey said. I'm a rising senior at Pomona College, and this summer I was working at the CCB doing analysis of autism causing mutations using data from the Simon Simplex collection. Um, so I'm going to start off by talking a bit about autism and why it's important that the Simons Foundation has devoted so many resources to studying what actually causes it. So it's a neurological developmental disorder, and it's caused by mutations that can either be inherited or occur spontaneously through like environmental factors. And the main thing with autism is that people who have it tend to um, it tends to impede like that ability to socially interact and communicate with other people. And as a reason, like the number of cases have been increasing every single year. And yeah, so that yeah. And then the Simon Simplex collection is a part of the Simons Foundation Autism Research Initiative, and it has a repository of genetic samples from 2,600 simplex families. And by simplex, I am referring to the fact that only one child in that family has autism, while their siblings were unaffected. The main things that I was looking at when I was analyzing this data, probans are what we call the children with autism in those simplex families. I didn't necessarily look at the unaffected siblings. I was looking at the genomic data of the people with autism in the families that we looked at. And then DNA and RNA disease impact score. A disease impact score is what we use to determine the likelihood of a certain mutation contributing to autism, and that can occur in like DNA and RNA. I'll talk more about the structures of the two later. And then gene symbol and genetic variation. So with every single gene, you have like in every DNA, you have a strand, and strands have base pairs. So whenever there's like the mutations that we're looking at was when like one base pair mutated to another one. And I'll talk more about that later because it's also important to what I was looking at. And then distance to representative TSS. TSS stands for transcription starting site, which is where the first nucleotide binds to to RNA during DNA transcription. And then loss of function intolerance means the resistance that a gene has to losing its function once a mutation occurs. And I also looked at that. So my objective was to see how variance and TSS distance affect the disease impact score, whether certain variants and certain like which means like certain mutations were more likely to cause autism, and how far from the transcription setting distance would that like how far would the mutation have to be to also contribute to autism. And then transcript transition variants, whether they were more impactful than transvergent variants, and I'll talk more about that later as well. But a brief background about DNA replication. So this is a DNA double helix strand, and as you can see, there are cylinders. I don't know. It's a cartoon image meant to represent like a DNA strand. And as you can see, there are different colors binding to different colors. So the different colors represent nucleotides, and nucleotides have a complementary nucleotide that they bind to in the other DNA strand. And whenever there's a transcription, the RNA has to be complementary to the strand on which like it will bind to with the DNA once you open up the double helix structure. And then from there you can translate it, but that's not important for what I'm talking about. So as I mentioned, we have base pairs, we have purines and pyrimidines, depending on that structure, as you can see here. And the transitions occur when a purine changes to another purine. So like adenine in this case would mutate to guanine, or cytosine to thymine. And these are less impactful because like purines bind to pyrimidine. So whenever a purine mutates to a guanine, it doesn't change as much because it will still have a pure like a complementary pyrimidine or guanine to bind to. And as you can see from the structure, we have like the NH and OH bonds, and they bond together using like through like the OH or NH bond. So to talk about the DNA disease impact scores, that's what I was focusing on. I was trying to see whether there was a discrepancy between like certain mutations and that disease impact score. And as you can see, they generally follow the same like Gaussian structure. So despite the fact that we didn't have like the numbers were vastly different, like as you can see for the the green ones here from TG to the gray one, which is TC, the number of mutations were vastly different from like 2,000 to 10,000. But generally, the spread was the same. I centered the mean at 0. So there wasn't a lot to tell from this, which is why I resorted to heat maps. And from heat maps, I wanted to see if there was a correlation between the disease impact scores of like when a gene like when the base pairs mutates from like CT and like GA. So I split them apart from transition to transversion, and here you can see that I use like covariant matrices. So the black ones, which are on the lowest scale, represent correlations that aren't as strong as the ones like as you go up the scales, like the strongest correlation would be white. So the thing is, for every single base pair that I was looking at, the strongest correlations that they had was to each other, but in the reverse order. So in this case, like a CT's strongest correlation was to TC, and then TC, and then CT, and then GA, AG, and then AG and GA. But when I reviewed the other base pairs, like they generally had the same correlation. And that also showed in the transversion variant heat map, where the other, as like if for other base pairs, the correlation wasn't as strong. And I included this to show it in a much lesser, because I can see like this, it might be a bit more like a lot to look at. So this is just an AC variant map. So we have like AC and CA, and then AG and GA, and it follows the same pattern. So from there, I looked at the analysis of the disease impact score and the TSS distance for both DNA and RNA. And from here, you can see that like, even as the TSS increases, the disease impact score, it doesn't increase as much. So there's no like positive or negative correlation to see. And the same thing is also for RNA disease impact score. And the thing that I'm planning to do going forward is to to try to figure out why the way like the correlations aren't quite the same, because you can see like from the RNA graph is a bit more spread out, and then from the DNA graph it's a bit more on the left hand side, so it follows a bit more like an upward increase. And then from there, I also wanted to see how the RNA disease impact score and the DNA disease impact score correlate. And there's absolutely no correlation. As you can see, like it's really just spread out all over the place. And then I mentioned loss of function intolerance. And this is also going to be part of the future work where this is the loss of function for all genes. It didn't necessarily look at the variants. So as I go further with this project, I want to focus on the loss of function intolerance for each specific variant and see how that correlates. And then I was working with SCC data. For future work, I plan to work with Spock data, which is a bit more extensive because it will be featuring 50,000 people, 50,000 subjects. This one was only at 2,600. And I will also be able to analyze both like simplex and multiplex families. So simplex, like multiplex, meaning that multiple people in the family have autism. The phenotypic data and the genomic data as well, because for this one, I was only looking at genomic data. And I also intend on employing a bit more a cluster analysis. So yeah, with that, I'd like to thank my mentor, Shirley, Natalie, for her advice, Casey, Saran, and Shannon for organizing this whole thing, and then NSBP and the Simons Foundation. Thank you.
Great. Are there questions for Refilwe? Yeah, Carmen? Yeah, press the black circle. It'll pop up and you'll see a green light.
Oh, okay. I had a question. So I noticed your work was looking at missense mutations. I was wondering like what your data would look like if you looked at like another type of mutation, like a point mutation. Do you know?
I know because I was working with already existing data,
Okay.
so I wasn't part of the data collection.
Oh, okay.
Yeah, but I think that is something worth investigating in the future. But right now I'm just working with data that has already been provided to me.
I see. Thank you.
So actually, since you have this slide up, I have a question. When when you know looking at the bottom plot for the loss of function, it seems like it's pretty flat, but then when you get to 1, it just skyrockets. And so I guess could you just give a physical understanding for why it is that right at 1.0 the number of
Yeah,
mutations just skyrockets?
that is something that I do want to investigate, because disclaimer, I don't have a good biology background. So like for the most part, I was really just trying to see if there were any correlations between like the variance and the disease impact score. But loss of function intolerance is something that I really want to look at. So that is a good question for me.
Okay, great. Are there any questions on Zoom? All right, I'm not hearing any. So let's thank Refilwe again.